Good morning, my dear students. So we have to continue our discussions with midterm topic. We have now the enzyme structure, classification, and mechanism of actions. So in this topic, we're going to discuss the um, structure, no, of the enzymes. And classifications, how they are classified according to the chemical reactions they undergone, and of course based on their MOA or mechanism of actions during the uh, chemical reactions between the substrate, and then we have the enzymes. Okay, so learning objectives: by the end of the lecture, the students should able to, number one, define enzymes and the related terms. So we'll describe the active site, okay, the apo enzymes, the holo enzymes, the prosthetic group, the enzyme specificity, okay, and explain the energy of activations. Okay, take note that during the chemical reactions, energy is being used up by the enzymes in the process of catalysis or speeding up the reactions. Know the mechanism of actions, and then you have to explain the classification of enzymes, and of course to describe the structure of enzymes. So importance: the enzymes plays an important role in metabolism, okay, like digestion of food or digestion of biomolecules in the body, or for example, the conversion of your sucrose into two monosaccharides. So sucrose is undergoes hydrolysis process that will be converted to fructose and uh, glucose acted by enzyme sucrase. Okay, and of course the hydrolysis of proteins that is acted by protease enzyme, or the hydrolysis of fats that is acted by lipase enzyme. So it is also used in the diagnosis, okay, in laboratory diagnosis of the different diseases, and of course in the therapeutics. Okay, so there is what we call importance of the enzyme, wherein there are a lot of enzymes that are inhibited by a drug, okay, to prevent the certain abnormal conditions of our body. All biochemical reactions are enzyme catalyzed in the living cells. Okay, example, the breakdown, of a protein that is considered to be enzymatic reactions. The breakdown of carbohydrates is also enzymatic reactions. That's why in the breakdown of glycolysis, there are a lot of enzymes that plays the important role. This example is step one, the hexokinase enzyme, wherein glucose is converted to glucose 6-phosphate that is acted by hexokinase. For example, it's a good indicator in a disease such as myocardial infarction. Okay, uh, we'll discuss also the diagnostic importance of the enzymes if there is what we call a higher level of your uh, as arginine, aspartate, or enzyme that is involved in the specific diseases. So enzymes can be used therapeutically, such as digestive enzymes also. So define enzymes. Enzymes are proteins that increase the rate of reactions of lowering the energy of activation. So enzymes are proteins that speeds up reactions, okay, and there is lowering of energy upon activation, because the lowering of energy is due to the fact of the enzymatic reactions between the reactant and then the enzymes, okay, or between the substrate and then the enzymes. They catalyze nearly all chemical reactions taking place in the cells of our body, okay, not altered, consumed during reaction. So there is no alteration in enzymes, or they are consumed, but when enzymes binds to that substrate, they have irreversible reactions, and they are detached after the chemical reactions, and they are known to be reusable. Active sites, okay, enzymes molecules contain a special pocket of clef called the active sites. If we are the substrate, for example, glucose is the substrate, it binds with the active site. And then the substrates entering the active site of the enzyme. Okay, this is the enzyme, we have the active site. As long the substrate fits with that active site, enzyme changes the shape and slightly as substrate binds. Okay, enzyme, it will form the enzyme substrate complex. For the example that is glucose enzyme uh, complex, example, glucose lysokinase, and then the product, the enzymes and then the substrate undergoes chemical reactions, so enzyme product complex will form, and of course after that one, products leave the active site. So this is considered to be mechanism wherein enzymes are reusable. They are not altered or modified during and after the reaction process. So we go now to the LuckinKey theory. This explains enzymes specificity. Okay, specificity. I mean, okay. This explains the loss of activity when enzymes denature. So the denaturing of the enzymes depends upon the temperature, the pH. Okay, the LuckinKey theory, we have the the key is the substrate, and the lock is the enzyme. So when the key fits, okay, or it corrects fitting, or the key fit to that enzyme, then there is what we call reactions will follow. If incorrect, there is no reaction. So there is what we call lock and key. As long the substrate fits with that active site, reactions will follow and product will be produced. And take note, enzymes remains as the same as usual, and they are usable. So in the lock and key theory enzymes actions, take note, active site has a rigid shape. Only substrates with matching shape can fit with the enzyme. The substrate is the key that fits the lock of the active site. So once na nagbind dito po yung substrate, then it fits. So usually parang ina-unlock niya. So there will be chemical reactions that will happen. Okay. So ano naman yung tinatawag natin na working cofactors of the enzyme? So we have here the APO enzyme. Okay. So APO enzyme is the inactive part of the protein. Okay. We have the catalytic site, this one. Catalytic site or known as the active site. We have the APO enzyme known as inactive. We have the cofactor or the non-protein part, which plays the important role in the utilization of the substrate. And we have the holo enzyme known as the active enzyme. Manghambang ka ng active enzyme, mayroon siyang coenzyme, take note. And mayroon siyang cofactor. Okay. And of the same, it is a whole with the APO enzyme. So yung hatawag natong holo enzyme is known to be active enzyme. So this one, mga ka-active enzyme, mayroon siyang APO enzyme. At the same time, may cofactor, may coenzyme. That is that comprise, the active site, the active enzyme known as holo enzyme. Okay. Important terms of understanding of biochemical nature and activity of enzymes. We have the cofactor. When you say cofactor, it is a non-protein chemical compound that is bound either tightly or loosely to the enzyme and is required for catalysis. Usually, ang cofactor is function similar to the prosthetic group. Usually, the binding is transient, dissociable manner to the enzyme and the substrates such as ATP. Okay. The most common cofactor is magnesium and zinc. So, types of cofactors, we have the coenzyme. At the same time, we have the prosthetic group. Usually, ang prosthetic group, so, they functions for the utilization of the substrate, or they tend to facilitate the point of generation of the substrate to the active site. So, almost the same sila ng what call this one, cofactor, but yung binding ng prosthetic group is not transient, while yung cofactor is transient. Okay? Parang temporary yung cofactor. So, coenzyme, the non-protein component loosely bound to APO enzyme by non-covalent bonding. So, when say non-covalent, the bonding is considered to be there is no sharing of electrons between the coenzyme and then the APO enzyme. Okay? Examples, vitamins or compounds derived from vitamins or chemical moieties, okay, transported by coenzymes includes the methyl group, the acyl group, the oligosaccharide, and dolicol. For prosthetic group, that's non-protein components tightly bound to APO enzyme by covalent bond is called prosthetic group. So, The difference between cofactor, yung cofactor is transient, okay? While yung prosthetic group is considered to be covalent in bonding. That's why the bonding, I mean, of the prosthetic group to the apo enzyme is considered to be what we call this one, stable, okay, because of covalent bonding. Unlike for coenzyme that is non-covalent. So in covalent, there is a sharing between two molecules. So yung sharing between the prosthetic group and with the holoenzyme. Okay. So the enzyme specificity, we have enzymes have varying degrees of specificity for substrate, or the degree of the specific enzymes. So nagdepende rin siya sa specific na substrate. Okay? Enzymes may recognize and catalyze single substrate. Pwede isa lang. Example, single substrate, glucose lang siya. Okay? A group of similar substrate. Okay? Or group of similar substrate, or group of proteins, okay, or group of amino acids, okay, particular type of bond, okay, so depende sa particular bond, ano ganing yung particular type of bond. We have covalent bonding, non-covalent bonding, okay. So types of enzyme specificity, we have absolute, catalyze one type of reactions or single substrate. Usually ito yung urease, catalyzes only the hydrolysis of urea, kasi mga baka urease for urea. I mean, group catalyze the reactions of similar substrate. Example, hexokinase adds phosphate group to hexoses. Hexoses, I mean, or hexose. Mga baka hexose, these are fructose, glucose, the mannose, and then we have the galactose. So, specific na yung hexokinase mo. Hexokinase, tawag na to, similar substrate pala. Okay, pwede yung hexokinase to other hexoses. Okay. Linkage, catalyze one type of reactions, specific type of the bond. So, covalent lang. While example of this one is chymotrypsin, hydrolyze the hydrolysis of peptide bonds, or known to be the proteins also. So, specific na siya, specific type of bond. So, kung ang catalysis niya involves covalent, so covalent lang siya. Next, important terms to understand biochemical nature and activity of enzymes. The activation of energy or energy of activations. All chemical reactions require some amount of energy to get them started. So in every chemical reactions that takes place between the enzymes and the substrate, it requires some amount of energy. So it is first pushed to start the reactions, parang lock and key. Okay, energy is called activation energy. So there is what we call activation energy. So when enzyme is being activated, energy is also used up. So tawag doon activation energy na, because that activation energy will facilitate the chemical reactions that the enzyme and substrates will undergo. So mechanism of actions of enzymes. So enzymes increase reaction rate, decreasing the activation energy. Okay? So, once na-increase yung reaction, nag-decrease yung activation energy niya kasi gina-use up niya. So, enzyme-substrate interactions, there is a formation of the complex. So, meaning, when enzyme binds with substrate, it will form ES. That is the complex already. Okay? So, complex is also reversible. Okay? And the product of that complex and the reaction, okay, or the outcome of that complex is known to be the product. So, lock and key model. Okay, and we have the induced fit model. So, ni-induce siya, tapos it will fit to the active site. Okay, try to see the active site. That is induced fit model. And lock and key, so we have the key, and then i-unlock niya. That is already chemical reactions that happens. So, enzymes lowers ang reaction activation energy. So, without enzyme, take note. So, we have here lactose. So, lactose undergoes the hydrolysis process that is facilitated by enzyme or acted by enzyme lactase. Okay? So, net energy release from splitting of the lactose. Usually, magnubo yung energy mo. So, glucose plus galactose, that is a product of hydrolysis of lactose, which is acted by lactase. Activation of energy without enzyme. Okay, so this is the activation energy. So with enzyme, kung without enzyme, activation energy without enzyme mataas yan. With enzyme, kasi i-consume na, kasi ito wala. So with enzyme, so kasi wala pang lactase, lactase. So, kanina wala pang there is no presence of enzyme. This one is present of enzyme. Lactose is acted by lactase. Okay, this is the enzyme, this is your substrate. Then the product is glucose and galactose. So activation with energy that is lower, and net energy is released. Kasi dito wala namang energy. So matagal. Try to say medyo steep yun, mataas yung reaction niya. Dito mabilis, and then there is what we call lowering of an energy level and net energy is being released. So yan ang kinalain ng enzymatic reaction lowers activation energy without enzyme and with enzyme. So to correct, kaya wala pa yung lactase dyan. Okay? To correct. Okay, this is the lactase. So makita nyo, hindi madali lang yung reactions. Ito medyo mataas, dugay siya because there is no enzyme and there is no consumption of energy. So once nag-bind na yung lactase and lactose, so this is your substrate, so energy is being lowered, and then the release of net energy release already. Okay, so we have a free energy and we have the progress energy. So based on that illustration kagina, so course of reactions without enzyme, if try to see, that is considered to be steep, mataas. Okay, so without enzyme, so the energy is considered to be intact, and if there is an enzyme, so reactants, ito yung reactants mo mataas, so the reaction is considered to be slower, and course of reaction with enzymes, so makita nyo nubo na siya, this is the red one, it will form the product. So there is what we call the energy is unaffected by the enzyme, so EA with enzyme is considered to be lower. So walang changes yan. Okay, yung mga energy level considered to be unaffected yan. Okay, this is considered to be energy. So yun lang ang kinalain nila, basta mag-use once na merong enzyme, mabilis yung reactions, while without enzyme mabagal. Okay, so this is the progress of the reactions against the free energy. So LuckinKey model, so we have the active site, so it will form ES or enzyme substrate complex. So LuckinKey model, so as what we have discussed. So in the LuckinKey model of the enzyme actions, this active site has a rigid shape. Okay, so usually kailangan ma-fit yung substrate mo. Only substrates with matching shape can fit, and then substrate is a key that fits the lock of the active site. Okay, and then this is an older model, however does not work for all enzymes, so it depends upon the chemical reactions. So induced sweet model, we have the induced sweet model of the enzyme actions. Active site is flexible, not rigid. So usually parang hindi siya mag-fit. Okay, i-induce mo lang siya para mag-fit siya. Okay, the shapes of the enzyme active site and substrate adjust to maximize the fit. Okay, maximize the fit. Okay, which improves catalysis. There is a greater range of substrate specificity. So meaning yung specificity ng substrate mo, kahit na ganito kalaki yung active site niya, so kailangan there is adjustment of the substrate to maximize the fit based on the active site. Then induced sweet model, the enzyme plus substrate, then it will lead to enzyme substrate complex. This model is more consistent with a wider range of enzymes because yung yung active site hindi naman talaga parehas talaga sa shape, okay, ng substrate. So kailangan magkaroon ng tatawag na i-induce siya para what we call this one, i-induce na ng konti para magkaroon ng tinatawag natin na what we call this one, fitting of the substrate with the enzyme. Next, enzyme substrate complex, step 1. So enzymes and substrate complex to form E plus S, it will form ES. So enzyme, and we have the substrate complex. So the reaction is reversible. So try to see if yes, the enzyme, and then we have here the substrate, then it will form a enzyme substrate complex. Okay. So next, step 2, enzyme product complex. An enzyme product complex is formed. So we have ES. So usually that is reversible reaction. So you have enzyme product complex. So we have your ES. Kagina yung ES is your enzyme substrate complex. Then undergone reactions. It will have the transition state, then it will form the enzyme product complex. So, reversible pa rin yung reactions. The enzyme and the product, usually, EP can be separated. E plus P, so pwede pa rin yung natawag natin na reversible reactions. So, enzyme product, then we have reversible reactions. So, the enzyme is ready for another substrate. So, usually, after that one, product will be released. Okay? So, what affects enzyme activity? Number 1, 3 factors yan. We have environmental, cofactor, and coenzymes, enzyme inhibitors. So, yung environmental mo depends upon the temperature, okay, the pH. Cofactors mo, coenzymes, so this depends upon the cofactors that are being used up. So, enzyme inhibitors, so there is what we call different substrate competitors or inhibitors during the chemical reaction process. So, environmental conditions, so ano yun? Extreme temperature are the most dangerous because enzymes are proteins, they are heat level. So, usually, yung higher na lang na maximum na temperature na pwede i-attain na enzyme is almost 40 down to 60 degrees. So, depende yan or 70 degrees. Depende upon. After that one, enzymes will denatured. So, high temperature will denature or unfold the enzymes because all enzymes are proteins and proteins are very sensitive to temperature, higher temperature. So, pH 6 to 8, nearly neutral lang. Pag sumubra sa doon sa 8, denatured na yung enzymes mo. And it depends upon the substrate concentrations. If there are lot of substrate concentrations, mas faster yung enzymatic activity. If konti lang yung substrate concentration, so the tendency medyo medyo madugay-dugay ang substrate, ang tawag nito, ang bonding. Kasi kung damo, mas dasig mag-bond. Okay? If damo ang competitor na naman, yun ang problema. Okay? Pero kung substrate na siya, walang problema, maband mo lang na siya. Okay? Cofactors and coenzymes, we have inorganic substances. Yung zinc, vitamins, respectively, are sometimes need for proper enzymatic activity. Example, iron must present in quaternary structure hemoglobin in order to fit with pick up oxygen. So, usually, mga iron, usually bind with quaternary. So, hemoglobin is based on the protein structure that is quaternary. So, optimum temperature, take note. So, nasa human body, optimum temperature is 40. Okay, it depends. So, turnover or in thousands per minute, usually, pwede na siya sa 40 degrees. Okay, 40 to 60. Pero usually, kung 60, hindi na. Kung sa body lang class, optimum temperature to correct, that is 40 degrees Celsius. After that one, denatured na yung effect ng enzymes. Next, pH environmental factors. pH also affects the rate of enzyme substrate complex. So, most enzymes have optimum pH of 7. However, some prefer acidic or basic. So, we have pepsin. Okay. So, we have the low or natawag natin turnover number nila. So, acids yung pepsin mo because it is found in the stomach. Or the trypsin. Okay. So, kung alkaline yung trypsin mo, usually human amylase enzyme. So, neutral with alkaline. So, depending upon the environment na present yung enzyme. Okay. What will this one? The different enzymes that are present in the intestine. So, ano na yun? What we call this one, they are already alkaline. So, with enzyme kinetics. The study of the rate at which an enzyme works is called enzyme kinetics, wherein there is what we call involvement of energy in the form of heat. Okay? So, enzymes are protein catalysts that all catalyze speeds up the rate of chemical reactions without being used up in the process. So, usually, mayroon lang siyang energy but hindi na-consume yung enzyme, hindi siya na-use up, hindi rin siya na-degrade. They achieve their effect of temporary binding to the substrate, and doing so lowering the activation energy needed to convert it to product. Okay, so take note, their efficacy usually depends upon the binding with the substrate. So it depends upon the binding, o anong bonding doon, covalent, that is strong bond, okay, or weak bond siya, or weak bond ba siya, okay, van der Waals forces ba siya yung pag-bond niya, okay. So the rate at which the enzyme works is influenced by several factors. So we have the concentration of substrate molecules. The more of them, the quicker the enzyme molecules to collide and bind with it. So basta damo yung substrate gas, that's why I told you a while ago, the faster is the bonding or binding or colliding. The concentrating substrate is designated as S, express the units of molarity. Okay, so molarity yung unit. As the temperature rises, molecular motions, hence collisions between enzyme and substrate speed up. Take note, so malabot na nila yung 40 degrees to succeeding sina class, pwede mag-denature enzymes. Yun ang maximum temperature niya. Okay? But as enzymes are proteins, there is an upper limit beyond which the enzyme becomes denatured and ineffective. Usually, yung pinaka-minimum is 40. So, sometimes, mayroon yung chemical nature ng enzymes, pwede sa sumaka sa 60 or 70, but mostly 40 degrees lang yung maximum. Okay? So, the presence of inhibitors. Competitive inhibitors are molecules that bind to the same active site. Okay? As the substrate preventing the substrate, from binding as they do so, but are not changed by the enzyme. So, may tawag natin inhibitors. So, there are a lot of inhibitors in the chemical reaction. So, they compete with the substrate in binding with the enzymes. Okay? Non-competitive inhibitors are molecules that binds to some or other site of the enzymes, reducing catalytic power. So, kumbaga, itong mga non-competitive, ito yung humahawak sa enzyme, or ito yung kung sa ilong ginhawiran yung enzyme. Okay? So, once na hawid sa doon sa other parts ng enzyme, so the enzyme has tendency to lower its catalytic reactions in binding with the substrate kasi mayroong nag-bond doon sa other site ng enzyme. So, nagtawag ng non-competitive, meaning wala siya compete sa substrate. While yung mga competitive, they compete with the substrate. So, for example, we have here, plotting VI as a function of your substrate, we find at low values of S. Okay? The initial velocity, or VI, this is your VI, okay, rises linearly with increasing substrate. So, kung ano yung tawag natin initial velocity, kung increasing yung substrate mo, nagasaka rin yung velocity, correct? But as S increases, the gains of VI level up, forming the hyperbolic reaction. Ito yung tawag, hyperbola. O rectangular hyperbola na siya na what we call this one, the tawag ng hyperbola mo, pasaka. Okay? O rectangular hyperbola graph. The asymptote represents the maximum velocity reaction known as your B max. Ito yung B max, yung pinaka-maximum velocity niya. Okay? As the enzyme, what we call this one class, basta yung enzyme mo naga-react siya, so nagasaka man yung maximum velocity. So nagadepende pa rin yan sa concentration ng substrate. Okay? So the substrate concentration that produce is VI is 1/2 Bmax, is designated as Michaelis-Menten constant known as Km. Okay? So Km is equals to 1/2 Bmax. Alter scientists who developed the study of enzyme kinetics is known to be Michaelis-Menten. So, VI, initial velocity, that is moles per time, take note. While S is the substrate concentration. Okay? So, kung damo yung substrate mo, nagasaka pa rin yung speed mo. Correct? Bmax is the maximum velocity that the enzymes initiate or produces. Km, substrate, is known to be Vi is 1/2 of your Bmax. That is your Km. That is your Michaelis-Menten constant. Okay? So, initially, we have here the VI represented by A, B, and C. Okay? So, your C is the Bmax. Your B is 1/2 Bmax. That is 1/2 of the what we call this one of the velocity. So, Bmax is divided by 2. Correct? This is divided by 2 kasi ito yung maximum mo na Bmax. Okay? And A is considered to be, kinatawag nato na refers to the velocity also of this one. Okay? So, depende yan kung mag-extra ka ng data mo. So, kukuha, ito yung parang initial, ano nito, initial speed mo, yung A. And then, yung Vmax mo sa kalate, and yung C mo, ito yung pinaka-maximum. Okay? So, Michaelis-Menten equation illustrates that mathematical terms, the relationship between initial reaction, velocity, that is, initial velocity is Vi, substrate is concentration. When S is much lesser than Km, point A, S, so, ang S mo lesser sa Km mo, tama? Or 1/2 Bmax, lesser siya, A. Okay. And your term K plus S is essential equal to Km. So, replacing the K plus M with Km reduces the equation. So, kumbaga, yung Vi, it is the standard formula, Vmax times S, then over Km plus S. So, manamba ka Km, that is 1/2 Vmax. So, to replace class, okay, to simplify, I mean, the reaction, so i-divide mo siya, so pwede ma-cancel yung S. So, Vi equals to Vmax times S over Km. And or Vi initial, so, is equals to Vmax over Km times S. So, same lang yung, approximately pares-parehas lang yung pag-simplify ng reactions. Okay? So, when S is much greater than Km, so point C mo, mataas ang yung C. Okay? That is the maximum velocity. The term Km plus S is essential to S, equals to S. So, ang gawa niyan, replacement of Km plus S with S reduces the equation 2. So, ito yung natawag natin na standard formula. So, meaning, yung Km plus S mo is equals to S, ha? So, Km plus S is equals to S. So, S nito, pwede mo i-cancel.